Thank you very much. I'm conscious I don't have a particularly loud voice. Can you hear me if I speak at this volume at the back? Yes? Okay. Please, if I do need to move the microphone or change my voice, then let me know. It's a great pleasure and a privilege to be here and to speak at such a prestigious institute. I would have apologised for bringing the rain from the UK, but from what Representative Hardcastle was saying earlier, it's a good thing that we brought the rain with us. I would like to make some general observations about international stem cell policy to begin. I used to have a presentation with a map of the world indicating what different countries did in relation to embryonic stem cell research. But as I was saying to Kirsten earlier, almost on a weekly basis that would go out of date. And it does become very difficult to provide that sort of overview. And it can also be a little dry if you're just comparing one country to another. So I'm going to try to give an overview without going into too many specifics. I then want to look at how policy has developed in the UK. Again, as Rob mentioned, Lord Patel is going to tell that story in detail tonight, and I don't want to spoil that and give you the ending. But I want to give just a few pointers on that. And then I want to look again at the EU, how Europe has influenced policy development and regulation in the UK. If you look at how different countries across the globe have regulated embryonic stem cell research, and again I'm going to focus for the purposes of this section of the talk on embryonic stem cells rather than adult and other types of stem cells because of the complex policy and ethical issues. If you look at how countries have regulated, it's possible to summarize about 4 different categories of regulatory models. There are those countries like Germany and Italy who've introduced very restrictive, in some areas prohibitive regulation. In Germany, for example, the 1990 Act and the 2002 Stem Cell Act prohibit the creation of embryonic stem cell lines within Germany and research using those lines, although they do allow the import of lines from outside Germany. Initially, the 2002 Act said it had to be prior to 2001, and that time frame has now been relaxed, but it's still very, very restrictive, and it's part of the German criminal law, the penal code. So if you break that law, then you risk going to prison. Italy, likewise, has a very restrictive policy on embryo research and assisted reproduction, which doesn't allow the creation of embryos that are not going to be transferred to a woman. And if embryos are created for the purposes of assisted reproduction, they have to be put into the woman. They can't be kept in storage, and they can't be used for research purposes. So those are 2 examples, and there are many more of very restrictive legislative frameworks and regulatory models that we see. Further down the scale, then, there are countries like the UK, and to that list you can add some of the Australian states, and also Canada has done the same, that have said, well, we are going to allow embryo research, we're going to allow embryonic stem cell research, but we're going to subject it to regulation. And we're only going to allow that research to take place with a license that's been granted by a government regulator. And the Canadian, Australian and UK models are very similar. In fact, they're largely based on the same principles. And we'll come on to talk a little bit more about that as well. Then there are countries, and again there are a number of these, although the number is dwindling, where there's no legislative model or framework, but there is guidance or professional self-regulation, which has been introduced either by professional bodies or by the government. And China is a good example of this, and also a very important example of this, because China has a huge amount of stem cell research that's ongoing at the moment, a lot of which is wonderful, and a lot of which is perhaps below par. And I spent a couple of weeks traveling around meeting stem cell researchers in China earlier this year, and all of them were crying out for regulation along the lines of what we have in the UK, because they desperately want that benchmark, the quality assurance that comes with conducting research such as that they're doing under a license and subject to scrutiny of the kind that we have in the UK. Then there are countries, and I think we have to include the US in this, where there is no national regulation. There is state regulation, but there's nothing. And this leads me to include one of my favourite quotes from the President's Council on Bioethics from 2003. The only workable approach found has been the policy of federal neutrality, whereby the federal government does not prohibit embryo research, but also does not officially condone it, encourage it, or support it with public funds. Now, obviously, that last clause in the paragraph has changed with President Obama's change in policy, but otherwise that I think still represents the federal position in the United States. Now, leaving aside those four categories, in between, on the one hand, complete prohibition or tight restriction, and on the other hand, no regulation, you have a huge array of different types of controls and restrictions and mechanisms that regulate research and embryonic stem cell research in a number of different ways. And I've just put a few up here, but there are many, many more. In the UK, for example, certain procedures are prohibited. We've already heard that reproductive cloning is not allowed. Conditions can be placed on the permission that is given to conduct research, and in the UK there are a wide range of conditions that are attached to every license that is granted for embryo research, and then additional conditions peculiar to the particular research that's being conducted can be added by the regulator. The purposes for which research may be licensed or conducted can be restricted, can be limited by statute or by government policy. And the US is a good example of a country where funding is a form of regulation. The restrictions on funding and the use of public funds for embryo research is a good example of a way in which, although not the traditional orthodox way of regulating, it is a form of regulation because it restricts the ability of universities that rely on public funding to conduct research. We've heard about limitations on the duration of research in the UK and many other countries. The 14-day limit has been adopted and accepted as the appropriate duration for which embryos should be allowed to be kept and developed and cultured for research purposes. And then we also see a number of different governance frameworks, such as the ones in the UK that Rob has talked about already. But you would expect to find ethics and governance oversight in any country where research of this kind is permitted but subject to regulation. But despite the wide variety of models and the wide variety of approaches that have been adopted across the world, it is possible to, if you look at them, to identify what I think are three key rationales that run through all of them, even if the outcome of considering these rationales is to ban everything. In all of the countries where there have been parliamentary debates and policy considerations for the regulation of assisted reproduction and embryo research, you see these three common themes coming out. The first, which isn't really relevant to our discussion this afternoon, is the welfare of the child that might be born as a result of assisted reproduction, and also the implications that assisted reproduction has for the family and the family model. The second, which is relevant to our discussion today, is public health and safety that might follow either from assisted conception or from the use of embryo research and products that might be derived from human embryonic stem cell lines. But the third, and the one that we keep coming back to, it's an issue that arises across the globe, but is obviously a key issue for Texas and the rest of the United States, but particularly Texas, is the status of the human embryo. Any country that has chosen to regulate and to legislate in this area has accepted that there is a special status attached to the human embryo. I'm not aware of any country where there is regulation or guidance or professional self-regulation where it's agreed that the embryo has no special status. There's a consensus that there is a special status, but there is no consensus, nor will there ever be, as to how that status should be protected or what that status really is. And that, I think, is where the real problem arises. The consequence of that is that if you're going to regulate, if you choose to regulate, there has to be a compromise. Because for those who believe that the embryo has a sacred status, that it is the same as a child or an adult, then any research involving that embryo is unacceptable. So any permission to conduct research involving human embryos, that particularly that which results in the destruction of the embryo, will be unacceptable. So there has to be a compromise to allow regulation to emerge. And in the UK, we've achieved that. And in other countries, it's been done. it is still unacceptable to many, and I want to emphasize that we are by no means through the difficulties and the debates that you're having here in Texas, in the UK. This is an ongoing issue, and I'll come on to talk a little bit about that in a while. It's also been mentioned today, and Rob said this earlier, that I think the UK, like a number of other European countries, is perceived and often referred to internationally as a permissive environment for embryo research and stem cell research. And this is quite typical of that. This is a quote from a researcher who was part of a group that looked at stem cell research in China and the UK as part of a large funding program over the last few years. And one of their most recent reports included this line, that European regulation is getting more permissive over time. And part of the material, the data that Thomas used to base this conclusion was the fact that in 2001, only 3 countries in Europe had legislation which permitted stem cell research. But by 2007, this had increased to 14, which permitted research, of which 4 also explicitly permitted somatic cell nuclear transfer. Now, it's true that there has been a change. It's true that over time, more countries have adopted a regulatory framework where these treatments and research procedures are permitted. What I would emphasize there, and it's not the first time you've heard it, and I don't apologize for saying this again, is that in the UK, this research is very, very tightly regulated. As an attorney and a litigator, I make a living out of representing universities and researchers who feel that it's too restrictive because they're not allowed to do what they need to do. And it is by no means an easy feat to obtain a license from the regulator to do research involving human embryos in the UK. And I wouldn't, as a result of that, describe our framework as permissive. It's potentially facilitative and potentially permissive, but you've got a lot of hoops to jump through before you're granted permission to do that research. So briefly, I want to make 3 points about policy development in the UK. Lord Patel is going to tell this story more elegantly and eloquently than I could this evening, and I want to just make 3 very brief points about things that I think are relevant to the discussion that we've been having today. After the birth of the first IVF baby in 1978, Louise Brown, IVF enjoyed a pretty much unfettered expansion in North America, in Europe, and particularly in Australia. And it was only really in the early 80s that politicians started to turn their attention, not so much to IVF, but to the regulation of embryo research, which at the time was very closely entwined. I don't want to go into the details of the Warnock report or the Green and White papers, other than to say this, that when the first report came out, the Warnock report, that recommended the sort of regulatory model that we have in the UK, it was very badly received, both by scientists and in Parliament. And it was so poorly received that shortly afterwards, those who were fundamentally opposed to embryo research and to this sort of framework introduced a bill in Parliament. A Conservative politician called Enoch Powell introduced the Unborn Children Protection Bill, which would have prohibited outright any research involving human embryos and would have placed significant restrictions on IVF. Now, this bill got through the first stage in the British Parliament and it almost became law. It was very, very narrowly defeated on essentially a technicality. And I mention this simply to make the point that in the UK, we very nearly banned embryo research. Now, this came as a wake-up call to the scientific community, and this is really the point that I want to make. Up until that point, there was no real lobby group on behalf of embryo researchers and those doing the sort of genetics research that was also part of this discussion at the time. And as a result of this close shave with the Powell bill, a body called Progress was a charitable organisation set up to promote a better understanding of reproductive and genetic technologies. This was set up to lobby parliamentarians and to make sure that people understood why this research was important and why it was important to allow scientists to use embryos, and they were very much the precursor of the sort of lobby groups that we see now. But had it not been for that, I think that there was a very real danger at this time that the research wouldn't have been permitted. The second point that I want to make about the story, if you like, is that I've mentioned here that in 2003 there was a case the Quinta Valley case against the Secretary of State for Health. Josephine Quinta Valley is a very well-known and highly regarded opponent of embryo research and assisted reproduction related research. I say highly regarded because she is she's very popular amongst those who agree with her, and she's very vocal and will always turn up to meetings such as this to make her views known and heard. And she has brought a series of cases which have challenged the regulator's authority to license in this area. And I think it's important to mention as a part of this story because the legal framework that we have and the regulators' decisions as to what they should and shouldn't license in terms of embryo research in the UK has been subject to regular public and political scrutiny, but also legal scrutiny through the courts. And the Court of Appeal in this case agreed eventually that embryos that were created through cell nuclear replacement were embryos for the purposes of the Human Fertilization and Embryology Act, and that the HFEA, the regulator, could issue a license for this research to take place. And that was a very important point in the history of the HFEA, and it gave them a lot of, it cemented their power and their authority to license in this area. But I think it's worth mentioning that in addition to the public scrutiny and the debates that we've had, it's also been something that's gone through the courts on numerous occasions over the last 16, 17 years. We've also, as Rob mentioned, recently reviewed the regulatory model and the policy that we have in this area in the UK. But the government decided that they wouldn't change the model of regulation, that the HFEA, as Rob has outlined, should remain. That it's been tweaked, and there have been some notable changes in what can be permitted, and the Act has been future-proofed, if you like, to the best of the government's ability to make sure that it can cope with the rapid progress of science in this area. But the model that we have is still considered, and this comes back to the question earlier about would we change anything, it's still considered to be the best way of regulating embryo research in the UK. And I wasn't asked the question, but I'll answer it. I would stand by that. And if the opportunity did come around again to think about how we would do this, I would still advocate this model. And I think that's why it's been adopted in Australia and why it's also exactly the same in Canada, because it works. It's a very good way of ensuring that there is scrutiny and monitoring of the research that's being carried out, but it's also a facilitative, permissive environment subject to that scrutiny. So yes, it's a potentially permissive environment, but it's subject to tight regulation and ongoing oversight. I should also add that the responsibility of the HFEA in this regard doesn't end when the license is granted. If you're doing embryo research, you have to report back to the HFEA at regular intervals to tell them what you're doing and how the research is going. And they can suspend that license if they don't like what you're doing. They can revoke the license if you do something that you're not meant to do. So it's not something that ends with the granting of the license. It's an ongoing process. The 1990 Act, but as it's amended by the 2008 Act, is now good for research. The research community, the scientific community, by and large, accepted last year that the new legislation that we have was a good thing. It was designed very much with progress in mind and with the developments that we've seen over recent years in mind to make sure that we don't need to go back to Parliament next year to say, well, now we've got to change the law again. And that is another important message, that if you are going to regulate in this area, you need to do it in a way that allows progress to happen and allows change to be taken into account. We do, as again Rob has mentioned, have public and private funding available for stem cell research in the UK. And we're lucky, I think, that we have government and public support for research and therapies derived from research in the UK, which is a privilege. So, in summary, as far as the UK policy situation today is concerned, we have an Act which protects patients through its public health and safety requirements. It allays public concerns by making sure that there is scrutiny and oversight of what's allowed. It protects researchers. It's a moot point among some, but the fact that there is a regulator that has to make a decision about what should be allowed does act as a buffer, if you like, from those who are opposed to the research and would criticise them for doing the research in the first place. And it also provides an environment within which innovation and progress can flourish. What I would say, though, is that if you're to regulate on this sort of model, you also have to take into account the 4 bullet points at the bottom. The regulator has to be adaptable. The model has to be adaptable. It has to be able to change and accommodate change. The regulator has to be proportionate in the way that they implement that regulation, because heavy-handed regulation, even within a potentially permissive environment, can cripple research and innovation. The regulator has to be impartial and objective in what they do and can't be subject to bias or intimidation. And there has to be expertise and efficiency on the part of those doing the regulating. Expertise is difficult because if you have got somebody who really understands this research and is very good at it, why are they going to stop doing it to go and work for the regulatory body? So there are issues to be worked through in terms of how you manage that. Now, I want to finish just with some very brief points about European law. When I was a trainee solicitor many years ago, I had a supervisor who told me that when you're giving a presentation to an audience, you should never apologize for anything that you're going to say. And I've always stuck by that principle and that rule, and I never have, and I won't today. I will simply say that European law is very dry. And there's not much that I can do to spice it up other than by using a bit of colour. This is an illustration of the tiers of regulation that one would have to address and consider and abide by in the UK if one were to take a tissue or cell-based product from bench to market. I don't expect that you can read all of the writing. You don't really need to. It's more for the visual impact of the numerous tiers of regulation that we have. And this takes into account both the national regulation that we have, but also the European regulation. There's a lot of it. We like to regulate in the UK. In Europe, our enthusiasm for regulation, though, pales into insignificance compared to Brussels and their passion for regulation. And so there is an awful lot of it. And I wanted to just talk about 2 of those, but before I do so, I have to just very briefly explain 2 principles, subsidiarity and direct effect. Subsidiarity comes back to a point that was made earlier, actually, about the way that the federal system works in the US. It's essentially the idea that the community shall take action, that's the European community, Brussels, only if and insofar as the objectives of the proposed action cannot be sufficiently achieved by the member states and can therefore, by reason of the scale or effects of the proposed action, be better achieved by the community. You can tell that was written by a lawyer. A good example of this is public health. So almost all responsibility and competence for public health is dealt with locally by member states. But there are some exceptions where the European Union intervenes to deal with issues which are of an EU significance, and 2 of those are cross-border health threats and so-called health tourism. I don't like the expression health tourism, but I think you'll understand what I mean, that it's where patients go to another country across a border in order to receive a treatment that either isn't available at home or they prefer to be treated elsewhere. The second principle is that of direct effect. It essentially means that certain European laws, if they fulfill certain criteria, have direct effect in member states. Whether or not that member state implements them properly or at all, they will still be directly effective and binding on those member states. And the 2 pieces of legislation I want to talk about are both directly effective in this way. So whether or not the UK had implemented them and whether other countries do, they still have effect. And here we see, this is just a snapshot of the European Commission's website on public health. And if you have very good eyesight, you can see that the second bullet point at the bottom is all about tissues and cells. And this is where the Commission has a limited but important, notable remit to regulate at an EU level what we can and can't do. That's wrong. It's not what we can and can't do, but how we use tissues and cells. That's an important slip, actually, for reasons which I'll come on to explain. The first, in brief, that I wanted to talk about is a directive that Rob has already mentioned. It's the directive on setting standards of quality and safety for the donation, procurement, testing, processing, preservation, storage and distribution of human tissues and cells. This was introduced, as the title suggests, to make sure that wherever a patient went to receive treatment in the European Union, they would be assured that the quality and safety of the cells or the tissue-based, cell-based therapy that they received would be of a certain minimum standard. It explicitly applies to stem cells and to embryonic stem cell material, as is set out in the preamble. But it only applies to materials that are intended for human application. So it doesn't apply to in vitro research. It's only for that which is intended to be used in humans. It doesn't apply to autologous treatments. It doesn't apply to blood or organs. But in the subsequent two daughter directives that followed the main directive, there's an awful lot of information about the consent that you have to obtain from patients, how tissues and cells have to be packaged and labelled, import and export. I won't go into the details of these, but the technical directives are very, very prescriptive in terms of what has to be done to maintain those basic quality and safety levels. But it is precisely that. It's a minimum level of quality and safety. What the EU cannot do is require member states to permit embryonic stem cell research or therapy using embryonic stem cells. But if a state does permit that research and the development of a therapy to take place, and if states want to use those therapies, then they have to fulfill these minimum criteria. So it's often referred to as an umbrella law. It's not really. It's better described, I think, as a foundation. If you're going to permit the use of cell-based therapies, then you've got to build on that foundation. You can go further, you can be more restrictive if you like, you can even ban it, but you have to comply with these basic minimum standards. And I think it's an interesting analogy with the US position, and I don't know, I'm not an expert in US law as to whether or not that's a sort of model that could be applied in the US, but it's worked very well and has been quite well received in the UK. The second regulation that I want to discuss very briefly, it's a very complex piece of law, is the Advanced Therapies Medicinal Products Regulation. This was introduced by the European Commission specifically to deal with the sort of treatments and therapies that was the subject of this morning's scientific meeting. We already have well-established regulations for medical devices and well-established regulations for pills and medicinal products. But there was a grey area or gap, a lacuna, in between the two for the three types of therapies that are in the bracket here: tissue-engineered therapies, cell therapies, and gene therapies. And so what the Advanced Therapies Medicinal Products Regulation did was facilitate the introduction of those sort of therapies to the market with standards that were relevant to therapies derived from, for example, stem cells. But here we see spelt out in explicit terms this issue that I've already said, that the EU cannot require states to permit this sort of work. So the preamble says the regulation of advanced therapy medicinal products at community level should not interfere with decisions made by member states on whether to allow the use of any specific type of human cells, such as embryonic stem cells or animal cells. It shouldn't affect the application of national legislation prohibiting or restricting the sale, supply, or use of medicinal products consisting of, or derived from such cells. So again, Germany is free to prohibit the development of therapies from embryonic stem cells or the use of embryonic stem cells in Germany, and there's nothing that Brussels can do about that. But in the UK, we have to comply with the standards that are passed down. But again, the ATMP is good. It's been well received. It provides one single EU marketing authorization route for people who want to take a therapy to market and to introduce it to patients across Europe. It brings us more into line with the US, Canada and Australia, and it's intended to facilitate access and the development of therapies without putting people off. I will skip this because Rob has already dealt with this. The consequence of these regulations is that we have, in addition to the HFEA, responsibilities passed on to two other regulators, the HTA, and then to take a product to market, the MHRA. And they have different roles and responsibilities, which maybe we can talk about at questions if you're interested. In terms of challenges, again, it's more for questions and discussion. I don't think we should be relaxed. The fact that we've recently reintroduced helpful, positive legislation doesn't mean that we should sit back on our laurels. There are interesting figures about how there are more conservative parliamentarians who voted against this bill than there were back in 1989 on the previous bill. So it's not something that we should sit back and forget about. But crucially, I think in addition to parliamentarians, we need to make sure that the media and patient groups are kept involved in the discussion and kept informed and educated about what scientists are doing. The implications of health tourism are difficult to manage and difficult to regulate. And it's not something that we've had time to talk about this afternoon, but it's a key challenge, I think, to make sure that patients can't simply go elsewhere for unregulated treatment that is dangerous and has an adverse impact on the rest of the community. And then we need to make sure that we can maintain investment, support international collaboration, and make sure that if scientists do have a good idea, that they can actually take that to the clinic and start using it in treating patients. Thank you very much.
Our next speaker, as he indicated, is Dr. Mason. Chris Mason.
Okay, so good afternoon, and I have the pleasure for stepping in for Paul Simmons, who's unfortunately unwell. What I'm going to cover is a sort of 101 of regenerative medicine, with where the industry is today, and I'm going to look at all the various different cell types, tissue engineering, adult cells, and embryonic stem cells. I'm going to look at medical innovation. What are we actually dealing with stem cells? Why is it so very different? Looking at the whole approach of management of disease versus cure, talking about regenerative medicine as I've just discussed, and then looking at the past, present, and a little bit into the future. We all understand this. This is a gradual improvement. You can make a brighter and brighter candle, but you never make a light bulb. And this is very important when we think about the state of pharmaceuticals. We can make better drugs, and we made certainly better small molecules. Aspirin is great at treating headache. We then moved on to the biotech revolution, where we then had recombinant human proteins and monoclonal antibodies, and great drugs like Herceptin and insulin, but they're not a cure, they're a treatment. They manage the disease. They look after, reduce the pain level, control blood pressure, but they don't cure it. And what patients really want is they want a cure. They want to be returned back to normal. And that's what we have the opportunity of doing with regenerative medicine. So it is a step change. It's not a gradual increase, a little bit over small molecules, a little bit over biologicals. It's something entirely new, using cells, living cells, as therapies. And it replaces or regenerates human tissues and organs to restore or establish normal function, returning us back to normal, hopefully in one hit. So we use this term regenerative medicine, we use the term stem cells, we use them interchangeably. It's not quite right. Regenerative medicine, certainly tissue engineering, and I'll come back to that, making organs in the lab, using cells as therapies. But it also covers things like regenerative compounds, things like bone morphogenic protein, for example, for repairing bones, are very successful. EPO, erythropoietin, for increasing red blood cell levels, is regenerative medicine. And also, we have an ability to put scaffolds in the body, inert material, to help regeneration. Now, I'm going to just deal with tissue engineering and cell therapy, but those all four are part of the whole sphere of regenerative medicine. I put this one up. Everyone knows who those characters are in the room. They all had incurable diseases. None of those diseases were treatable. None of them were curable. Spinal cord injury, Parkinson's, Alzheimer's. And there is a large unmet clinical need out there that we're all very aware of. We also see a vast amount of information about what the scientists are doing. And we now have a very good science base on which to move forward. And 300 patients have already been treated with living cell therapies. That's really important. Even students in my own group at UCL are not always aware of that. So we're talking about 300 people out there today with living cell therapies. There are 4 FDA-approved products, 1 cartilage and 3 skin. Look at the date they were done by. It was 1997 to 2001. We're not talking about something particularly new, but we just don't seem to be aware of how successful it has been. So, what are these living cell therapies? And what makes them different from a biopharmaceutical? This is Lego. I'm sure you have it in the US. We certainly do in the UK. And you can build things out of it. This is the building blocks. It's equivalent to the cells of our body. And this is a Lego man. But this is how we are. We consist of a series of cells put together. It's no good giving small molecules when you need to replace actual cells. And so we have cell therapy. Could be adult, could be embryonic, and IPS has all been mentioned this afternoon, and I'll give some examples. Or it could be tissue engineering, where we combine the cells with a scaffold. All equally important. And we've had much discussion today about embryonic stem cells. And we have an embryo on one side of the screen going towards, through normal development, going towards the adult human. And at every step, there are cells which are useful to us for therapies. Embryonic stem cells, adult cells, and we heard about umbilical cells and a host of others. So we have this broad spectrum of cells available to us. No one is going to cure all. We need a mixture of these different types of cells to get the results we need. If we structure them in the order that they've historically been used, adult cells, that's what 99.99% of you and I are constructed of, adult stem cells next, and then embryonic stem cells. If we look about potential for therapeutic use, I think there is no doubt the embryonic stem cell has the most potential. At the same time, it is the most scientifically challenging, and we heard about iPS cells. This is the converting of adult cells into embryonic stem cell-like cells is even more scientifically challenging. And then we have the increasing difficulty of commercializing embryonic stem cells. However, underlying that is the potential for their therapeutic use. But I'm going to cover all these cell types. All have a value. So, I'm going to start off with an example. 10 years ago, tissue-engineered skin got approved by the FDA for use for chronic ulcers. This is a piece of tissue-engineered skin in its package. It's white because it hasn't got the pigmented layers that we have within our own pigmented cells we have within normal skin, but it has an epidermis and a dermis. And here's a typical leg ulcer that it's used to treat with good efficacy and safety. And something like 250,000 patients have received Apligraf from Organogenesis, and here it is being used to treat a leg ulcer. Likewise, there is a company called Tengian who are tissue engineering bladder. So they take material from the patient, take cells from the patient, grow it up, put it onto a structure that resembles a bladder, basically a balloon. And here we have one being grown in the dish in their laboratories. This is a bladder being grown in culture media. And this is one actually being popped into a patient. They have treated patients. They're in phase 3 clinical trials. They've shown safety. They've shown some degree of efficacy. And they're now on the home straight towards getting full FDA approval. So here's the bladder being popped in. And here's the bladder in place with the ureters on it as well. So we are achieving success with regenerative medicine. There's absolutely no doubt. But what about stem cells? And what makes a stem cell particularly important? Why is it so exciting? Well, a stem cell can make more stem cells, and it can make specialized cells. And we've all seen this picture many times, that the embryonics can make all the cells of the body. Adult stem cells can make less cell types. So mesenchymal stem cells, for example, are very good for cartilage, bone, and fat. But if you really want to make nerves, heart, and the like, you probably need an embryonic stem cell. And here's obvious what this is. This is embryonic stem cells turned into heart. It automatically beats of its own accord. But do we know if they can be cures? I've shown you those good examples of bladder and tissue-engineered skin. We know that stem cells are the building blocks of life. We know we've got animal data. But what about in man? We've got these different types. Adult stem cell, bone marrow stem cell transplants. We've all heard of those and the fact that they've done immense good for tens of thousands of cancer patients. And I'm going to talk about another one, which is using cells to restore eyesight. Bone marrow, very commonly used, and yet initially it was very difficult. Hundreds of patients died before they perfected the technique. Today, 500 patients have benefited from limbal cell therapy. This is an example of a person who's received an injury, a chemical injury to the eye due to cement dust, which is very alkaline, burning the front layer of the eye off, and then burning the stem cells underneath that would normally replenish the surface of the eye. So blind, very painful, and normally treated by taking the eye out for the pain and popping in a glass eye. Treating signs and symptoms, not regenerative medicine. We can take stem cells from the good eye. They reside at the 6 and 12 o'clock position. It's very simple. We just take a small biopsy, about 1 to 2 millimeters square, from just below the coloured part of the eye here. So very carefully taking out the stem cells. We then dissociate that material, grow the stem cells up and put them on the inside of a contact lens. You can't inject the stem cells on the eye or they just wash off. So we take the contact lens, have the cells grown on the inside. The surgeon very carefully cuts the scar tissue off the damaged eye. It's stitched on, the contact lens is stitched onto the damaged eye with a layer taken off. The stem cells migrate across, and in 80% of patients, they go back to a totally normal vision and normal looking eye. 500 of those being treated worldwide, 20 by Julie Daniels' group at the Institute of Ophthalmology, Moorfields Eye Hospital in London. So this is an adult stem cell therapy that's got safety, got efficacy, works today. 500 patients are better off for it. What about embryonic stem cells? Well, we've got lab and animal data, yes. And we've heard about Geron today, initially getting a phase 1 approval for spinal cord injury, and then getting put on clinical hold more recently due to an increase in the number of cysts, little sacs of fluid seen in the ongoing animal studies, which they've needed to continue with. What are they trying to treat? Well, this is a scan, a section through a human, and you can see a complete intact spinal cord. Unfortunately, I don't know if I have what to do. Okay, so we have here is the actual vertebra here, and the long white strip down here is the actual nerves travelling down. This is a damaged one. Here we see the vertebra, and here we see the nerves going down. And what actually happens is that it's not instant paralysis. What happens is that it gets crushed. It crushes the spinal cord, and the outer cells, the actual insulating cells, die first. And then the neurons die. And if you can interrupt the process whereby the insulating cells don't die, the neurons don't die. And that's the principle of the Geron therapy, that it disrupts the natural progression of the disease, prevents the insulating cells dying, and that stops the neurons themselves dying that run down. And the phase 1 clinical study was to look for safety. And we've still got to wait and see what happens there. But it's a study which has got a large amount of animal data. It's like 2,000 animals showing safety, and now we're moving forward, and obviously more data needs collecting. But this is the goal. Everyone knows who this is, and that's the result of a spinal cord injury. He did very well. He was extremely well looked after. He had a mind that was very powerful, but you can still see, on a ventilator, puffed out with his steroids, never walked again. What else is close to the clinic? I think someone else asked. Well, the closest ones probably are Novacell in San Diego with their insulin-dependent diabetes, are very close to sort of late-stage preclinical studies. Age-related macular degeneration, Rob mentioned. This is a disease that affects 25% of the over 65s that causes blindness due to loss of retinal pigment epithelial cells at the back of the eye. If you can restore those cells, you prevent the person going blind. And that's a project going on at the Institute of Ophthalmology with Pete Coffey in London and funded by Pfizer. And then heart failure. There's a group in Paris who are looking at embryonic stem cells for the treatment of post-heart attack to stop the heart becoming damaged and going towards getting heart failure. So we have a number of therapies that are moving forward in the clinic with good, strong animal data. So it's about gradual improvement. Yes, that was what we saw in the days of pharma and biotech. But we've moved on to stem cells with the promise of cure and not just to manage signs and symptoms. I think it's very important to realise that we are really moving towards a step change, and everything we've heard today about the regulation and the legal side is grappling with that step change. We're not just making a small step, a small little tiny change. We're making big changes. When you make big changes, you need to have the rules and the regulations in place to accommodate that. And biotech is very odd in that we get regulation before we ever get the therapies on the market. If you look at other industries like the internet, for example, the regulation follows once they're successful and they're generating money. We're around the other way. We are regulated before we've even got a product to market, and that doesn't help us move things forward quickly, but it's very important to have things which are safe and effective. So I leave you with just a couple of things. These therapies are very early, but millions of patients could potentially benefit. We're all agreed in this room. But I think the other bit of the equation is it's their families and carers, and there's millions of those as well, who will also benefit from stem cell therapies. And I believe that the 21st century is certainly the stem cell age. Thank you very much.
You might have a seat there with a personal discussion. Wow, if you weren't inspired with that, you're asleep. It's too late for you. So that was terrific. Thank you very much, both of you. And now we open for public participation. So the floor is open. Yes.
Is it correct that Ireland is the only country that really implemented in their law all those regulations that Texas proposed to be in 201? Namely, in their legislation, no stem cell research, no IVF, and no therapies that are even found in the future will not be allowed to use in Ireland. I don't know if that's correct, the statement. I heard it somewhere.
I don't think it is. I might be wrong on that, but I'm not. I know it's geographically very close in proximity to the UK. I'm afraid I don't know what the Irish position is.
There's plenty of IVF.
Yeah, there's plenty of IVF.
Yeah. I mean, certainly they're very strong on adult stem cells, and they've got a program with Smith and Nephew, I think it's called Remedi, which is looking at using adult stem cells for therapy, and that's very well advanced. But I can't comment on the other one.
It sounds wrong.
So how do you regulate people going across borders to get their health and their stem cell transplants? Medical tourism, as you tried to call it.
Oh, that's a topic we didn't cover today.
Well, it didn't cover, but James here talked about it and presented some information, and it's a very hard topic to discuss, and it's certainly, I can't imagine how you could even regulate that.
You can't, and the attempts that have been made to regulate it have been disastrous. Germany briefly tried to regulate people going abroad for terminations of pregnancy by carrying out gynecological examinations at the border, and you can imagine the response to that. I mean, it's impossible to do. We know very little about how much people travel for stem cell therapies. We do know there's empirical data about how much people travel for pre-implantation genetic diagnosis, which is another controversial treatment which is prohibited in a number of countries. And we know that within Europe alone, thousands of people move around Europe for PGD in other countries. And then usually it's because it's prohibited in their home country. And we know that they're not prepared for treatment, and we know that they're not followed up when they get home because it's prohibited. And I think there's every reason to believe that the same problems would associate with those who go abroad for stem cell therapies, that if it's prohibited, then doctors are not going to want to be involved. And that's a problem. It's a problem for the patients, and it's a problem for the doctors as well.
I think the other thing is that we have to move forward. The fact is that stem cell research has come a long way, both in the UK and US, and we are great at the research. Where we're falling down is really the translation. How do we turn these into safe, effective and affordable therapies that can be deployed for routine practice? And we need to drive that forward. Now, that takes years, because it's quite right that we go through the clinical trials process, get all the regulations in place, make sure they are safe, make sure we've got reimbursement, the teams are trained. And in the meantime, what we need to do is to make material available to potential patients, their carers and relatives, so that they're informed of the potential risks and benefits they may be taking by going abroad. And organisations such as the ISCCR are now considering preparing, for example, checklists for people to be able to use, so they can ask the pertinent questions of the people doing the therapy, of the clinicians, of the healthcare centre, such that they can make a proper informed choice.
Will those therapies done overseas being a proof of concept to be able to implement those therapies in the US or in UK, for example?
You would hope so, wouldn't you? I mean, there's just because something isn't regulated doesn't mean it doesn't work. I mean, that is a problem for us. I mean, we can't say just because there's no clinical trial that it does not work. You know, most of the older small molecule drugs we have did not have clinical trials. And things like aspirin, you know, I certainly know they work when I take them for my headache. So what we have to do is make patients or potential patients and their carers and relatives aware of the risks they are taking.
Professor Mason, one of the comments that we all remember Dr. Michael DeBakey making was one way to sell embryonic stem cell research to the world is one medical breakthrough. But it sounds from your presentation we might be getting close. And the eye demonstration here, renewing the vision for the fellow that his eye burned out, would be very convincing to the public. I haven't seen much of that, and I think we're remiss in not showing more successes. Would you like to comment on that?
Yeah, I think you're absolutely right. I mean, I think we are very poor. We get very focused on embryonic stem cells, and we are not good about talking about the successes we've had with other therapies. For example, at Bristol and Barcelona, they prepared a windpipe, a trachea for a Spanish lady about a year ago. Now, we had good cover in the UK press. I don't know if it came to the US, but this was a lady who basically had TB, damaged one of the airways to one of her lungs, and she would have lost a lung and possibly her life. And the group at Bristol, in collaboration with the group in Barcelona, actually tissue engineered an airway for this lady, and she is doing well. She's back to normal, in fact.
Yes.
Yeah. I'd like to follow up on that with just a sort of a comparison to Texas. I hope you'll find this a compliment, Laura Patel, but several times in the past 6 years, we've actually drafted legislation that we filed in Texas that is modeled after the legislation that you were instrumental in passing in Great Britain. So we certainly know that the design that you've come up with is an excellent one in terms of regulation. You talked earlier about the need for scientists to work hand in hand with legislators in order to make these things happen. And I think one of the challenges, and I alluded to it in my presentation earlier, is that there are some very good reasons that it's exceedingly difficult for our scientists to talk directly. I mean, we had a premier, well-regarded leader in the Texas Senate who lives in the community of a major university in Texas who was not aware that embryonic stem cell research is happening in Texas. So how do you get around that and so that the information is available to legislators so that they can make good decisions, and that they realize the impact, particularly the economic impact in Texas of some of the decisions that we have come this close to making?
I think because looking at me,
I jumped away from the panel. I'm sorry,
but I don't want to take the thunder away.
No, please go for it.
But I think a lot what Chris said is correct. That the legislation should be an enabling legislation for science to work, but at the same time protect the citizens from science that is not relevant or science that could do harm. So the point I made earlier on was therefore, before the legislation is drafted, there has to be a partnership understanding the citizens' need, the scientists' need, and the legislator's role, which should be an enabling role. We are lucky in a way, and James showed you the algorithm of how things happen, but at that time, the debate, how the legislator got educated, the debate was at a very high level, following the birth of Louise Brown. Then headlines, I still remember, I still keep them, and I have PowerPoints to show it, that said, monsters will now be created in test tubes. Just the same as heart transplant, that happened earlier on.
Right.
And therefore, the level of debate in the public and the politicians and the scientists was quite high. The similar thing, in a way to a lesser degree, but has the same kind of possibility of harm occurring, is this so-called personalized medicine, the genomics medicine. And we've just produced a report that maps out how Britain should go through the process of implementing genomics medicine so that it does not lead to harm to citizens. Now, we haven't legislated yet, but the threats for legislation is already described in that report. Now, the in vitro fertilization produced an opportunity for us to do that. By the way, Lady Warnock, Baroness Warnock, as she is now, it just so happened that she produced a report, and soon after that she became a baroness. So she drove the legislation through the House of Lords. So that was of a benefit, and she's becoming more and more liberal in her old age. That's all. Because normally when we get older, we go the other way. So that's That in brief is the answer why it happened.
I think it's also true that we'd learnt our lesson on GM food as well. I think that was certainly part of it. And I think rather than being reactive, which is how the scientists had rather poorly behaved in GM, we've been very proactive, getting to know the press, getting to know the politicians, and therefore very much getting the public on side with us before breakthroughs necessarily happen.
But how do you, if a major source of your funding is the same entity that is considering these policies, it makes you quite vulnerable.
I think the fact is that we have funding from a number of different sources in the UK. Certainly some does come from research councils. Ultimately it all comes from government, or majority comes from government, but we have the research councils, but we also have commercially organised government departments, such as what was the Department of Trade and Industry, now the Technology Strategy Board, which is also helping fund the whole translation, the next step. Then we have Wellcome Trust, philanthropists, and I think it's a number of different routes that provides the funding for us to move forward.
Thank you.
I believe Nina Brown had her hand up.
I have a comment and kind of a question. I've lived with Parkinson's for 24 years, so I'm one of those that would travel anywhere in the world to have find a cure for it. But I also, or proper treatment. I also understand, though, having spoken with a number of scientists, that as we're talking about the adult stem cell research and the IPS, that none of this would have happened had it not been for embryonic stem cell research promoting the discovery of IPS, for instance. And I think it's important that maintain the knowledge, that people maintain that knowledge or know that. But I'm also concerned that Texas doesn't realize the economic impact that not going forward with not just banning it, but not going forward with research on it would have to the state economically.
That's a very good point. I mean, I personally feel this very strongly about the UK. I mean, stem cells are both health and wealth. We're out to improve patients, out to improve the healthcare system, but we also need to actually produce a whole new regenerative medicine industry for these new therapies. And it is the last trench, I like to think of it, in the war in health. We've got small molecules, which did a great job, which mainly is big pharmaceutical companies. We then had biotech, which was pretty much invented in the UK and commercialized highly successfully in the US, mainly in the sort of Boston area and the West Coast. And now we move on to the final sort of challenge, the final method of moving forward, which is using whole cells. And there is no doubt that companies that successfully commercialize this will build a very competitive and sustainable industry in the future. Texas, like the UK, are well positioned to do this, but it does need the investment specifically in the translation of the basic science into therapies for routine clinical practice.
Well, considering that we're sitting with the biggest medical center in the world,
you're well positioned.
I completely agree with everything that Chris has said. The only thing I would add, and it's not to detract from that, it's simply a word of caution, that when you engage with politicians, you must avoid being dragged into the debate on the commercialization of embryos.
But it's not just embryos, is it?
Oh, no. Yeah. But it's a very, very difficult area that adds another layer of problems to the discussion.
Question back.
Yes, I actually have two questions. What's pluripotent stem cells? Where does that fit into this talk? And at the same time, where's the real convergence of nano-engineered materials and stem cells?
Okay, so pluripotent stem cells are cells which are capable of forming the 200 different cell types of the human body. The first pluripotent stem cell we have is the embryonic stem cells, and the embryonic stem cell-like cells, these are these induced pluripotent stem cells, whereby influencing 4 genes, we're able to reprogram the normal cells, say skin cells of our body. Both are pluripotent stem cells. It just means that we can make them in vast quantities, and we can make all the different cell types of the body. So I think when we talk about pluripotent, we're really talking about embryonic in terms of therapies. We've got 10 years worth of data suggesting that they are going to be useful in the clinic. Lord Patel quite rightly recognised the fact that iPS cells have still got a long way to go. They're at least 10 years behind, but they're also much more complicated in that the way that the genes are switched on or off is closely linked to how some cancers are formed. So we need to have a much better understanding of the science. What we need to do is to drive forward with somatic cells, adult stem cells, embryonic stem cells, and iPS cells.
Thank you.
I would just like to bring to bear some information about state funding. I'm with the Gulf Coast Regional Center dealing with the Texas Emerging Technology Fund, and they have been bringing forward dollars to the universities in a collaborative process with also a commercial entity. So Texas A&M University and Scott and White have been funded for a regenerative center, and I'm sure they were very careful on the terming of what type of material was in that proposal. I would expect that it's probably scaffolding, regenerative adult, etc. There's also one for the Children's Regenerative Center that's in consideration right now, the Texas Medical Center. And also we have a trauma center that was also funded through the Texas Emerging Technology Fund, which may bear some research eventually for solutions, and again with probably careful wording. The Cancer Fund, the $3 billion Cancer Fund, they're creating the foundation for the agency. They have RFAs that are out there right now for some of the basic dollars going out, and they're still trying to determine what the box is. But I know MD Anderson is going to compete, and there's a lot of stem cell work being done there and throughout the medical center. So I think we're moving forward. They're just careful of the wording and the terms and trying to hold the line with respect to certain areas, but they are moving forward with these topics. I mean, the dollars and the state are moving forward. So I just wanted to get some information per some things that are going forward, but probably with very careful wording.
Well, I think you're right. But all those things feed into one another. They're not isolated. So cancer stem cells, work we do there, molecular biology, all these things feed into each other. And so that we get this body of knowledge to move forward regenerative medicine in the end. I think the other thing that's very important is that stem cells aren't just regenerative medicine. We can use them, for example, to look for new drugs and also for discovery for things like toxicology studies instead of animal studies. So there is a lot of interest in stem cells outside straight regenerative medicine for other applications.
Very good. Thank you.
From the policy side, I like the idea of making it some type of enabling legislation instead of restrictive legislation. But do we still try to sell? I mean, we are a country of our own, aside from being citizens of the United States of America, and we're bigger than UK. So, but do we try to sell everything that's been successful around the world to get the public on the side? Because you were talking about Lord Patel, you had to have public input and the scientist input and then the policymakers input, or the policymakers divide it up and make it fit. Did we go out and sell these MS treatments that they're doing in Pakistan and China and Mexico and Costa Rica and the eye that you showed in one of your slides?
If you want the public on board, you have to have therapies which are proven to work, I think is the important thing. And I'd like just to pick up on the medical tourism issue which was raised. Houston is immensely successful, I'm told, at this medical tourism game. 66,000 whatever healthcare professionals here. And an amazing industry. You imagined you had excellence in stem cells here in regenerative medicine, how much bigger it could be here. And I think that's the big challenge for you.
Well, and that's part of the reason it gave me my idea, because I've been in contact with a group all from the Metroplex area that have been to Costa Rica. Well, if you had 20,000 a pop on those 50 people, that's not chump change. I mean, it's scattered over a 2-year period, and it's not big numbers like scientists are used to looking at as far as budgets and stuff, but as far as money that was leaving the state that they could have been doing at MD Anderson or the Heart Hospital or Herman or somewhere, if we enabled it to happen faster.
Yes, I mean, that's definitely true. You have the potential, like we do in the UK, to build a new and sustainable industry. And you can split that industry into two components. There is the use of allogeneic cells, that's cells where you have a common donor, like embryonic stem cells, and they lend themselves to a product, to a commercial product that's scalable. The other end of the spectrum is taking the cells from a specific patient themselves, so-called autologous. That is much more service orientated. It's something you would embed in your own existing healthcare centre. And so those two models need taking forward together. They're very separate in the way you move them forward, but combining the two together gives you a complete joined up industry. Sorry, yeah.
Would it be true to say that right now the science where it's going shows that every type of stem cell holds the promise
Absolutely.
of treating one or other disease?
Absolutely.
And the only thing that the embryonic stem cell holds the promise for is that it could probably treat diseases on a mass scale, as opposed to autologous one person. The limbal cell is a fantastic story, but it's one person.
Absolutely, yes. One person, yeah, that's right. I mean, we cannot get more than, well, these cells have to come from the patient themselves by and large, and we could not expand them up in numbers to treat them. Fortunately, it doesn't affect that many people. But if you wanted to tackle the big diseases like heart failure, stroke, insulin-dependent diabetes, Parkinson's, you simply would not have the ability to expand the cell types up. And this is where embryonic stem cells are very good. They're immortal cells. We can make them in, theoretically, unlimited number for therapy. Yes, Lord Patel's absolutely correct.
The other thing is Rob said that we are investing in hopefully producing 10 to 15, or at the most 25, clinical grade embryonic stem cell lines. But would you like to comment that if we had those lines of a clinical grade, well kept, would anybody else will need any other lines in the world?
I think we do need more lines. The big challenge with embryonic stem cell is that no two lines are the same, and that's been a real challenge for researchers. So it's possible to get a research grade line, which is exactly what Geron did with that H1 line, and they found they got great results in the animal model. Now, if they'd wished to go to a GMP, a clean made line, a newly made line, especially for therapy, it would have had different characteristics. It would have needed different conditions to differentiate it into the desired cell type. So it's very hard. No two are the same. And so if you're going to make a new therapy, whatever cell you really start out with is the one that probably is going to go through to the clinic in the end. So when we have these new GMP lines that Lord Patel's talking about, we will start in future with better, higher grade lines from the word go, and they'll make great starting material for our scientists.
James, you mentioned early on your presentation the struggle is with the status of the human embryo when it becomes the same as an adult or a child, and the struggle politically and everything. And you said that the UK had come to some, I don't know,
compromise.
Yeah, what was that? I mean, tell me how you got there, or because and you mentioned that if you believe one way, it's very hard to move them. Those that are in camps are in the camps.
Well, the compromise is to allow embryo research, but to subject it to regulation and licensing. So to allow research unrestrained, unrestricted in any way would be wholly unacceptable. But it was essentially a middle ground that said, well, okay, we will allow the research to take place, but we'll subject it to scrutiny, ongoing scrutiny, and you have to do that within a license that's granted by the government regulator. And that was the only way that it could really be taken forward. But right up until the law was passed, there was a question over whether or not embryo research would be permitted at all. And even the white paper that the government published had two options in it, one to ban it outright and one to allow it. But that compromise position is what's reflected in all of the countries where there's a regulatory framework that governs embryo research and stem cell research.
Okay, I just got the signal.
Could I just...
Yes, you have the last word. You have the very last word.
Okay, I'll get the last word. So, James, when you're talking about this regulatory structure, then are you talking about things like not allowing the process of paying women to produce eggs, and not allowing the fertilized eggs to differentiate longer than a certain number of days? Are you talking about those sorts of
One of my slides had a list of some of the ways and the mechanisms that are used to restrict what you can and can't do, and they can be very restrictive or very, very relaxed.
So you basically said we're going to allow the research to take place, but only in these certain circumstances.
Yeah, and they change.
For example, if they're fertilized eggs that were otherwise going to be discarded, then we're going to allow those to be utilized. And that's what you mean by compromise. We're not going to allow women to sell their eggs.
For example. Yeah.
Okay, I want to thank all the panel, of course, but I want to thank the audience too. Terrific. Thank you all for coming over across the pond and giving us the straight stuff.